Thank you all for coming back. So I am going to actually first figure out how this thing works. And I'm going to talk about some recent work that we've been doing in my lab related to an ongoing large project on the molecular causes of complications of preterm birth. So for those of you who've never thought about this, babies who are born early are, maybe not surprisingly, at higher risk of sort of every conceivable health consequence under the sun throughout their lifetimes, right? With, slightly elevated risk of everything. And we don't have a very good understanding of particularly why or what predisposes an individual to particular complications. And even within the period of time that they're in the hospital right after they're born. So mostly babies who are born early will stay in the hospital for some number of weeks or months until they're able to breathe on their own and eat on their own and stay warm so they can go home. During that time period, some of them have relatively uncomplicated clinical courses. Some of them get complications that put them at increased risk for lifelong illnesses and so on. And we don't really understand why that happens. The kind of standard approach from clinicians has been to focus a lot on infection as a cause of these disorders and complications. And that obviously plays a huge role, either infections they acquire in the hospital or infections that they, like maternal infections that cause them to deliver early in the first place. But that's not the whole story. It's becoming increasingly clear that there may also be genetic genotypes that predispose you to certain vulnerability that could be triggered by the insult of being born early and being on a respirator. Or maybe you have that same genotype and you weren't born early and you don't get some of these complications, but you're susceptible to things later in life for the same reason. So we're interested in looking at some of those connections. And because this is a network meeting, I'm going to focus on a particular project that we're doing that looks at network centrality in this context. I'm also going to talk a little about some temporal modeling stuff at the end, but mostly I'll stick to the network things. Right, and so this general concept, we're going to look at molecular networks, both protein interaction and regulatory networks. And this general concept of centrality is pretty well studied. There are lots of ways to sort of say how central a node is in a network. Right, so you can measure its distance to all the other, sort of average distance to everything else is sort of how central you are. You could look at how connected all of its neighbors are, right? But we're going to look at the notion of betweenness centrality, which is also a common method. Basically, it's just looking at, for any node, you're looking at the average number of shortest paths between all the other pairs of nodes that go through that node, and you normalize them by the number of pairs there are, right? So in a linear graph like this, for any node U and any other nodes V and W, there's only one path between them, right? So you're going to contribute either 0 or 1 to this sum. You normalize by all pairs of nodes, and you can kind of see that these values capture the notion of centrality that you might expect, at least in a graph like this. And they do so in larger graphs. What's more interesting is that it's been shown that this kind of centrality actually says something about function in molecular networks, right? So this is some work from about 10 years ago from Mark Gerstein's lab that basically demonstrated that nodes with high fractional betweenness, which he's calling bottlenecks, whether they have high degree or low degree, are more likely to be essential proteins in regulatory networks that are capturing information flow from place to place. So that he did not find the same result in protein-protein interaction networks, which I think is kind of interesting because it's sort of suggesting that the ways in which information moves in those networks may be a little bit different. Right, so this is old work, but it does show that this is a relevant concept. I should also point out, actually, that you could do this. There's a related notion to betweenness centrality called flow centrality, right? And you could look, instead of at shortest paths, you could look at all paths and look at flow. This work could be replicated with flow centrality and would probably be very similar. Our stuff could be replicated with flow centrality, would probably be very similar. We have not done that yet. So there's also a related notion of group centrality, right, which is just the average fractional betweenness centrality of all the nodes in a group. And this is, again, work from a number of years ago. What's interesting about that in the context of molecular networks is that you can now use group centrality to start to look at information moving through groups of proteins or genes. Right, so you can begin to say, well, okay, if I have, there we go. If I have a node here and a node here, and I'm looking at the shortest paths between them, there's one of length 4 that goes through this blue node, and one of length 4 that goes through this blue node. And so together, the two of these are capturing some notion of centrality for the shortest paths between those particular red nodes. And then, of course, you'd average over all pairs of possible red nodes in the graph. Right, so we actually want to take this a step further and use this to capture information flow in disease-related networks. So what does that really mean? Well, we spend a lot of time in our lab looking at expression data, but the problem with the gene expression data is that it tends to capture downstream effects of diseases, right? You're seeing maybe the consequence, genes that are expressed in consequence of some symptom of something that was caused by something else. We also have information, and growing amounts of information, about disease genes. Right? It's becoming easier and easier to do GWAS studies. People are doing lots of them, publishing this data, not necessarily replicating it. But so we have lots of disease genes, but they may not be differentially expressed. So the question is whether we can look at the flow of information from maybe a disease gene to a differentially expressed gene and find groups of things that are functionally related that are in some sense mediating this signal. And what can that tell us about disease? And so that's what we looked at doing. We're calling this pathway centrality. And we did all pairs shortest paths with these disease genes and differentially expressed genes in an experiment I'll show you. So the first thing I have to tell you about is what are the groups that we're using. There are some people doing work on finding or learning those groups, but that's another topic. We're just going to use set groups. We're going to start with Go biological process terms, because everybody does this. We're also going to use terms from the mammalian phenotype ontology. So basically, the mouse genome database has, I know everybody likes to work in yeast because it's really well studied, but we're looking at people, so mice are actually a better model. The mouse genome database has a lot of information about vast numbers of mouse mutants that have been characterized for particular phenotypes that the mutants exhibit, mapped to the mammalian phenotype ontology. Right? So we're going to say a gene is in the MPO phenotype term if there exists some mouse mutant that has, of that gene that has that phenotype. Right? And it may not be all mutants and it may not be all strains, and we're just not worrying about that right now. So a very simple way of pulling that together. And the data set we wanted to look at, we're particularly interested right now in a complication called bronchopulmonary dysplasia, which I will from now on call BPD. So basically, when babies are born early, if they're early enough, their lungs are not mature enough for them to breathe on their own. They are all on some kind of respiratory support. Right? And usually, they may start out on a ventilator and go to something that's just airway pressure and then go to oxygen. And eventually, hopefully by their due date, if not significantly earlier, they're breathing room air on their own. There is a subset of the population for whom this doesn't happen right. There is a developmental delay in the development of the respiratory system. Things don't go properly, and they don't get off of assisted reproductive therapy on schedule. That's sort of the definition of the diagnosis of BPD. So it's a very pragmatic definition. It's not molecularly based in any way. It's just like they're not on room air yet. But those infants who may even go home on oxygen often, again, grow up to have more respiratory problems. They're more likely to have asthma. They're likely to get early onset forms of emphysema. And we don't know why they get this. We don't know what's happening. So we decided to look at these three disorders from different life stages to see if there's some things in common. And again, we've seen some evidence in other places that there's some hints that there are some shared commonalities between them. Right? But asthma is something that affects people at all stages of life. COPD typically affects older people. And so we have expression data from blood in these cases and lung epithelial cells in these cases. We also wanted to make sure that pathways that are central in these data sets aren't just central in every network we look at. Right? So we picked a negative control. We had some data for Down syndrome lying around, and we didn't expect either inflammation or respiratory disease to figure prominently in Down syndrome, so we just used that. All the disease genes here are going to be the genes on chromosome 21, where you have an extra copy of those. And we have, and the expression data is from second trimester amniotic fluid. So I need to say a little about genes, particularly where we got them from and how they overlap. we have, from some previous work on disease genes, a data set looking at combining stuff from Huge as a database of basically curated GWAS results and OMIM. So we did that for BPD. When we did it for asthma and COPD, we got so many more genes, like 1,000 genes or something, because there are so many GWAS studies, and most of the stuff isn't reproducible. Fortunately, there are so many GWAS studies that there are reviews that focus on things that are reproducible, and so we picked those. So there are smaller sets from those. What I do want to say is there's a little bit of overlap in the gene sets, but by and large, the disease genes are pretty distinct. However, the differentially expressed genes are hugely distinct. And we use the same kind of methods and analysis for the microarray studies. COPD is RNA-seq, and we used whatever the authors used. Regarding networks, right, we took protein-protein interaction experimental data out of string and got a network of this size. We also did a regulatory network with some recent ENCODE data that's substantially fewer edges, although almost the same size node set. But one thing that's important is that in a directed network, reachability becomes an issue when you're talking about shortest paths. So actually, the network is noticeably smaller than in the protein-protein interaction case, and we'll see what impact that has on the results. And the last methods thing I want to tell you about is this concept of how do we know what high betweenness really is, what's meaningful, right? So we can compute a betweenness score. Our thought was to compare it to the betweenness score of a random gene set of the same size. And when we did that, this is kind of made up degree distribution data, but it captures the notion that I want to show, which is that if you're sampling randomly from something like this, you're going to get a huge number of very low degree genes, right? Our gene sets are basically functionally characterized genes that are actually biased towards having higher degree and being more central. And so initially, everything was significant. And that didn't seem very compelling. So we looked at some methods of sampling based on degree matching and so on. But the simplest way we found to address this is even easier to explain than all of that, and that gave us almost the same results, is looking at significance by sampling from the two core of the network. Right, so we're just going to find a maximal subgraph with a degree at least 2. We're going to do this by repeatedly removing nodes of degree less than 2. And we do that again and again until we can't do it anymore. And then we sample from this thing. So that gives us significance levels. And at the moment, these are not adjusted for multiple testing beyond that. So take the actual probabilities with a grain of salt. But at least the rankings are definitely consistent. Right, so what do we find? We actually find way too many results to summarize in here, right? There's a lot of stuff that comes out of this for all the different data sets. But the things that we see in common, I just want to give you some of the flavors of what we're finding, right? So the first thing we'd find out is that you do see sort of the kinds of inflammatory and immune system processes that you would expect to be implicated in all 3 of these pulmonary disorders. And you also see more specific pathways being implicated. I'm going to come back and talk about insulin receptor in a minute, but I think I won't go in. There's like a story for each of these lines, and I can't stand up here and tell you these stories all day, but I'll tell you a couple of them. Right, but we have, so we get some more specific mediating pathways. When we looked at the protein interaction networks with the gene ontology results, the initial thing we found was that there wasn't a lot significantly that was sort of below our arbitrary 0.05 threshold in all 3 disorders, except for very general terms involved in transcription. And most of those were also involved in Down syndrome, or significant in Down syndrome. That's not so surprising, because one of the things we know about Down syndrome is that the cells make a really valiant effort to tamp down the fact that they have an extra copy of that chromosome. So you don't actually get 1.5-fold upregulation of all of those genes. There's an effort to get back to normal. It doesn't quite succeed, but they're trying. So that, and by the way, on the previous slide, most of these, except for these 2, are not significant in the Down syndrome control. So I felt pretty good about that. But if you relax this criterion just a little bit, you start getting stuff out that I think is significant. And I think the reason for this, right, it's a lot more consistent with the protein-protein networks, with a few exceptions. But I think the reason for this is that we have a much smaller network in practice because of the directionality of it. So I just want to focus on a couple of results in here. One that I thought was interesting is response to hypoxia, because you can certainly imagine that babies born early who aren't breathing well or in a low oxygen environment, people with COPD potentially are not getting enough oxygen. People with asthma, if they're actually controlling it well, are probably not hypoxic, right? So this is probably an average of people who are and people who aren't. And you get something that's borderline significant, but not really. Down syndrome, though, this isn't just a false positive, right? This is one of the things we really know about this stage of development in Down syndrome is that oxidative stress is playing a major role. They're not able to cope with it properly. So I actually believe that's real. The other thing I wanted to mention, since we're looking for mediators, is NF-kappa B as a mediator. So this is known in asthma and COPD. It's actually suggested as a therapeutic target because it's known to be a mediator of inflammation in those diseases. It is not known in BPD. The only thing I could find that was at all suggestive is an animal model that sort of suggests that there is NF-kappa B mediated disruption of airway development in response to certain kinds of inflammation. Right, so it's possible. This is a hypothesis we're going to test in bronchopulmonary dysplasia. Let's see, in the interest of time, I'll just say that the mammalian phenotypes are a little bit more specific pathways, but pretty much giving us similar kinds of results. Interferon gamma is going to be of interest. So a couple of the new things that I wanted to focus on are this insulin receptor thing. We know insulin plays a role in asthma and COPD, right? We know obesity, diabetes are associated with these. In bronchopulmonary dysplasia, nobody's studied this stuff as much at the molecular level. What we do know about this, again, is that if you disrupt a certain insulin receptor signaling in rats, they get disordered airway development, right? So you can begin to see a possible pathway and maybe a possible treatment mechanism for bronchopulmonary dysplasia, if you know which individuals are likely to be able to be the ones to get it. Interferon gamma I'm also interested in because there actually is a particular SNP that's been associated with 2 of the 3 of these. The third one doesn't have anything about it in the literature. It hasn't, as far as I can tell, been tested. But we do have some collaborators who are looking very closely for variants that are associated with COPD, and they're very interested in this kind of stuff. So another thing that we can look at, right, so I was just doing Down syndrome as a control, but it turns out this is interesting in Down syndrome because we actually threw in way too many disease genes, right? All we really know about the disease genes is you have a whole extra chromosome. And people have been arguing in the literature for years about partial deletions and which parts of the chromosome are responsible for which of the Down syndrome phenotypes. We actually have a way of looking at this, right? So you can look at, let's say, this low muscle tone, right? We have no idea what on chromosome 21 gives people low muscle tone. But we can start to look at what are the pathways that are actually going through that set and see if we can figure out what genes are involved in that and what we could do about it. So the other major point that I wanted to make about all of this is that, right, again, you could look at each of these sets of results and tell stories all day. But one of the things that falls out of the bronchopulmonary dysplasia stuff is there seem to be a lot of gene sets involved in sort of developmental delays of various kinds. Right, so you can understand why there's impaired lung lobe morphogenesis. That makes pretty good sense. It's less clear. The vasculogenesis thing I'm going to come back to. There's a big debate about what role that plays in BPD. Why is there delayed bone ossification, right? This isn't a bone disease. Does this make any sense? It kind of does because it seems from other evidence that there is a general, or potentially there is a general developmental phenotype that predisposes infants to getting this complication. We know it's associated with being small for gestational age or short stature, right? We don't really understand quite why, but it seems like there is a developmental phenotype that's implicated here. So we're going to look into that in a minute. Right. I just want to sort of show you what some of these pictures look like. And this is a tiny fraction of what is really a huge hairball in cytoscape. But we filtered it down so you could kind of see this. And you can make edge weights proportional to the number of shortest paths that go between them. The pathway nodes in blue here Not all of them are high degree, right? Some of them are lower degree, and actually this is probably more high degree ones than you would normally see in a subset of the graph. I don't know, I'm still not sufficiently fluent in Cytoscape to slice these up as well as I should be, but you can certainly see how these could be mediating the signal between the disease nodes in red and the differentially expressed nodes in green.
So the pathway nodes are the nodes that you are predicting based on the centrality?
The pathway nodes are the ones that have high betweenness group centrality, significantly high betweenness group centrality.
Do they have different enrichments than the original differentially expressed genes? I mean, are you learning something new thanks to the centrality?
Yeah, you are learning which pathways are mediating this signal.
Okay, but your validation is based on functional enrichment, right?
No, my validation is based on experiments, right? So, well, we're getting to it. I mean, so the experiment, right? So we're still working on the experiments, but that's where it's coming from. No, so I'm not doing functional enrichment because these are pathways chosen to be, in a gene set. So they're going to be enriched for that gene set, right? This is how we're picking them.
Can you repeat what the role of the red and the green is in the formulation?
Yeah, so these are disease genes that are like GWAS-associated genes with disease, or some animal model predicts that they're disease-related, are the red ones. And the green ones, I know I shouldn't have used red and green, but actually it wasn't my choice. The green ones are differentially expressed in those data sets I was showing you.
Those are the inputs of your algorithm.
Yeah. Right, and the pathways are input as well, but we select the ones that are considered significant.
And you're looking not on general centrality, but centrality with respect to connecting the red and the green?
Connecting the red and the green, and pathway centrality of the group connecting the red and the green for shortest paths.
So you're looking for those blues that will connect green.
I'm looking for those blues that will connect those with each other, yeah. And that will do so more significantly than other things. And the point of that is to find mediators. And some of the stories I've been trying to sort of say are showing you that there is some suggested evidence in the literature confirming this. And we're doing experiments now to actually try to validate things more directly. So the thing I wanted to talk about in the last few minutes is that this temporal dysregulation, right, this developmental dysregulation, actually we can look at in a different way. So we have some different work in our lab that's been focused on looking at developmental dysregulation in expression, actually on a bunch of other data sets. So I'm just going to give you the tip of the iceberg on the bronchopulmonary dysplasia data. But basically, there are lots of algorithms out there for time series analysis of gene expression and finding things that are differentially expressed over time. Most of them have the sort of following paradigm, right? You take a gene, you take some control data, you fit a model of some kind to that gene's expression in the controls, you fit another model to that gene's expression in the disease, you compare the two models, and if they're different enough, you say the gene is differentially expressed, right? And there are any number of variations on this theme. We're looking at this in a slightly different way. We're building only one model, so it's an outlier detection approach. And we're not building a model of genes, we're actually building a model of age or time. Right, so the idea here is if there is a pathway that is regulated with development, then it might be possible for us to predict age from expression of the genes in that pathway. Right, so this is a set of genes related to a function. And we're going to try to predict age. If it works well in the controls, if we get a good model where the errors aren't too large, then we're going to use that same model to predict in the disease state. And again, for some of these pathways, it may be the same in the controls in the disease. And for some, this age-related developmental expression profile may be disrupted in disease. And so we're going to see this by basically measuring the probability of the errors in disease to errors in the controls. So we actually do this. We fit a normal distribution to these errors, which turns out to fit reasonably well, and we use it to predict probabilities in both cases. And again, we do significance assessment by scrambling the sample labels in kind of the obvious way. And this is actually FDR adjusted, so I'm pretty confident the things that show up as being significant are really significant. When you do this with bronchopulmonary dysplasia data, right? So before we were just looking at sort of either genes that are differentially expressed in BPD, no respect to time, and disease genes that have nothing to do with expression, and pathways that have nothing to do with either of those, right? But now we're going to look at the time series data as a way of predicting over time. And what we see is one of the top hits in BPD is vasculogenesis. Right? We can build a pretty good model of age in the controls, not such a good model. It's very dysregulated in the disease. Right? And one point I want to sort of emphasize here is that, yes, this is the same expression data set as the other one, but we're looking at it in a very different way. And the genes in this set, which comes from the gene ontology, and the genes in this set, which comes from the mammalian phenotype ontology, are 90% different. Right? So we're looking at actually pretty different sets of genes. It's not totally independent, but it's pretty independent. And it's a different way of looking at things. We see differential expression of vasculogenesis. We also see not as significant, but the bone ossification pathway. Again, very different gene sets, almost 92% different between the ossification one from the MPO and from this. Right? And so the last point I think I want to make about all of this is that this is starting to shed some light on the role of vasculogenesis in BPD. So there is this huge battle going on among neonatologists, basically looking at, right, it's known that there are problems in vascular development in bronchopulmonary dysplasia or in infants who get it. It is not known whether that's a cause or a consequence of the disorder. And people are slinging mud at each other back and forth in literature about this, right? But we're actually seeing some signs of this even in infants who are 5 days old. Now, you could still say that's a consequence, even though they can't be diagnosed with the disorder for another month or two. Maybe they have something going on and it's consequential. We're going to try to look. We are now getting data. We just did some RNA-seq on expression from newborns and cord blood, and we're going to try and validate this and several of the other hypotheses that seem to be held, supported from the literature in our own models. So that's kind of what we're doing with this. Again, this is, ongoing work. But I think the point is you can actually identify mediating pathways, some that have been validated elsewhere, using this notion of pathway centrality. And you can learn something about how disease works, and we've been finding things that people have suggested as therapeutic targets, so maybe we can find more of them. But verification absolutely is something that we have to continue to work on, and we're doing those experiments. And so the upshot of all of this is I do want to really thank everybody who was involved in this. The pathway centrality stuff was mostly work of Jisoo Park, who's one of my grad students. The temporal modeling thing is Michael Petrus, who's another of my grad students. I want to thank Ben and Lenore and anybody else who's in this room. And Diana Bianchi is the neonatal geneticist who leads a group called the Mother-Infant Research Institute at Tufts Medical Center, and there are several doctors there who have been working on, and fellows and various other people who have been working on this stuff. So I really want to thank them. None of this stuff is published yet. What is published is the work that it's built on, the annotation related to development, the disease and development work, and some work on anomaly detection. So I wanted to put those references up there as well. And thank you for listening.
So I didn't understand how you do the age prediction.
Yeah, okay, good. I glossed over that a little bit. Where did I do? There's the age prediction. Okay, so that's a good point, right? So this is some kind of predictive model. We actually looked at a whole bunch of predictive models, but relatively simple ones since we're looking for something linear. The one that seems to work well and not overfit is partial least squares regression. Right, so we build a regression. It's basically just a regression model.
Between each gene and
No, it's the set of genes in the pathway and age. Right, so I'm definitely glossing over a lot of stuff here, but So we have a whole bunch of genes in a pathway or a set, and we're going to use those as age predictors.
And then you average.
And we did try. No, we don't average. We just predict age for that that particular
So
patient.
what are you using the expression of these genes?
Yes, this is expression. Yep. Right, but I mean, in some cases, we can't predict age from a pathway at all, and then we're not going to do this, right? We're not going to consider. We don't even bother scoring its performance on the disease state if it doesn't do well in the controls, right? So there are lots of pathways that have nothing to do with whose expression is not related to development, and we don't care about them, right? We just care about the ones where it's good. Are there any disease cases where, or is there a disease where that pathway that we had a good model for suddenly breaks down, right? And this is something we've used in this anomaly detection paradigm, is this idea of finding, just learning from one-sided examples and then looking and seeing where that model fails. And that seems to have served us well here as well.
And what's the typical range of ages that you are getting?
Yeah, so in the actually, so this is, again, this is a bigger study that isn't as networky, so I didn't spend as much time on it. The BPD data, the ages are from about 25 weeks. I mean, basically from whatever the youngest sort of viability in the study was, up to about 32. Beyond about 32 weeks, they're not sufficiently premature to be likely to have these complications. So we don't include them. But we have also looked at this kind of model in kids with autism up to age 18 or so. We've looked at it actually in adults with Huntington's disease, and you can see age of onset and predict age even in cases where they haven't shown symptoms of the disease yet. You can identify if they're outliers, so we can use it for a lot of different things. But yeah.
So I think I have two questions. So one, so if I understand correctly, you're taking every gene in the pathway, and that gene gets some coefficient to predict age, and that's how you build the model. Is that right?
More or less. We're taking all of them together, and yeah.
And then a follow-up question: are there any cases where in the disease you can predict age, but in controls you're unable to?
That's a great question. I don't know if we've looked at that, but offhand, I don't think so. I have not seen anything that looks like that. We could look at that more systematically, but yeah. Typically breaks down what I would love to see, and that happens occasionally. Let's see if I can do this. Is to see just a real a line that just shifts, right? We're seeing things that are older or younger looking. It's usually not that clean. It usually goes from ordered to disordered more than ordered to shifted low. But we have only looked at a few data sets, so we'll keep looking. Yeah.
Is that age in weeks, or?
This is age in weeks. This is just gestational age, or I should say maybe weeks post-conception is the right way to call that, because they've been born. But it's age in weeks. Yeah. Yeah, and this is, again, this is sort of binned. This is data that's binned by age from, they give you a week. They don't give you a week and 3 days or something. So again, when you have, on the autism data set, we actually had a version of it where we had very fine resolution ages, but the version we're using to publish is the published version, and therefore doesn't have that data. And you can see, the discretization hurts you a little bit, but not that much.
Did you look through a lot of the different pathways just to see how likely you are to get some pattern like this at random?
Well, that's why we did the scrambles, right? So these are all, so we looked through all the different pathways, but we also compared them to cases where we scrambled the class labels. So we say this patient has BPD and this patient doesn't at random, and then build the same model, and yeah, you get nothing out when you do that.
There are no other pathways
that are significant? No. Right, I mean, so these are all compared to a bunch of runs of that and then adjusted for multiple testing. So these are pretty consistently more significant than that. Right, there are diseases where like there's just nothing, right? So if you look at a disease and there isn't a good model of development in the expression data, and I actually thought that would happen with the Huntington's data. I'm really surprised that we found something. But my student was just looking at it and said, "Oh, look what I found." So can happen. Yeah.
Okay. Let's thank Donna again.
Thank you.